Take it away.
Thank you for the introduction. And yeah, so my project is based around elucidating the impact of histone mutations inside of cancer. I've been working on this for the past couple of years. So today, I'm just going to be showing some of the results that we've recently gathered from this project. So for some background, hold on a second. Yeah, so for some background, histones are a group of yeah, let's close that really quickly. No problem. So histones are a group of intranuclear proteins, and they represent the main protein component of chromatin. And they can be divided into 5 different histone types, including H1, H2A, H2B, H3, and H4. And while all these different histone types are distinct, generally they have the same tertiary structure with an N-terminal tail and a core domain that's comprised of 3 alpha helices. Now, when 2 copies of each of the core histones, being H2A, H2B, H3, and H4 come together, they're able to form what is known as the histone octamer. And then when the histone octamer binds with 147 base pairs of DNA, it can form the nucleosome. And then when this also binds with linker histone H1 and 20 base pairs of linker DNA, the chromatosome can be formed. Now, the nucleosome and the chromatosome represent the fundamental subunits of chromatin from which all other larger structures are derived. So for example, nucleosomal arrays can be derived from nucleosomes and chromatosomes through internucleosomal and interchromatosomal interactions. And these can further scale on to create entire chromosomes as well. Now, because of this, histones are actually able to modulate access to DNA and also can modulate how various effector proteins actually associate with DNA as well. So they're very important for regulating access and to gene expression, DNA transcription, DNA repair, and so on. But rather than actually just being static barriers to DNA accessibility, histones are actually highly dynamic. And consequently, they can also act as an epigenetic platform in a couple of different manners. And one of those manners is through the use of what are known as variant histones. Now, histones come in 2 flavors. They can be canonical histones or variant histones. And variant histones differ from canonical histones in a few different ways. And the first way is through the expression timing of variant histones. Typically, canonical histones are generally restricted to being expressed inside of G1 and S phase, whereas variant histones can be expressed throughout the entire cell cycle, and consequently, they can be deposited into nucleosomes, replacing canonical histones at any point inside the cell cycle. And additionally, variant histones are also they have low, in comparison to canonical histones, they're also very structurally diverse as well, which can allow them to take on different functions. So in combination, actually, here's an example. So here we have a human H3 histone alignment, and here I've just highlighted a few of the different canonical histones. And we can see here that really, as far as their sequences go, these canonical histones only might vary by a couple of amino acids. When in comparison, variant histones can actually vary by several different amino acids, or in some cases with some specific variants, they could even have entirely different domains, entirely additional different domains. So in combination, being able to be expressed throughout the entire cell cycle and having all these different structural features, it can actually allow these histones to take on different functions at different points inside the cell cycle. Now, additionally, in addition to having variant histones, allowing histones to act as a epigenetic platform, histones can also be post-translation modified. So this is just the addition of chemical moieties, such as acetyl groups or methyl groups. And there are a few different effector proteins associated with these post-translation modifications. There are PTM readers, which are able to recognize these post-translational modification marks. There are PTM writers, which are able to deposit these PTM marks, and PTM erasers, which are able to remove them. And so the addition and removal of these different PTM marks are actually allows the histone to take on different functions, similar to how a variant histone would with their different structures as well. Now, histones, since they associate with DNA and they're able to modulate DNA accessibility, as well as modulate how different effector proteins interact with the genome, they're consequently highly conserved. And this is seen throughout evolution and on a few different levels of functional redundancy inside of the human genome as well. Now, I did mention that variant histones actually differ structurally a little bit from canonical histones. However, generally speaking, it's the overall structure of variant histones and canonical histones, regardless, are the same. They still have that N-terminal domain plus that core domain as well that's made of 3 different alpha helices. Additionally, the functional redundancy of histones can also be seen inside the histone genome. There's approximately, even though there's 4 different histone types, which are generally interchangeable, any protein inside of one histone type is generally interchangeable inside of the nucleosome. So for example, we might have, let's say, 3 different H3 histone proteins, and these can all be exchanged inside of the nucleosome, providing potentially different functions. But on the genetic level, there's actually several genes which can encode the exact same histone protein. Now, even though histones are incredibly important to maintain genomic fidelity and proper DNA processing, they're still prone to mutations. And in recent years, there have been a number of different mutations which have been observed inside of histone coding genes that have been shown to actually be able to drive cancer development. And there's a few different ways that histone mutations have been generally thought to be able to drive cancer development. First, they can occur at post-translational modification sites, so altering PTM reading, writing, and erasing. They can also occur at core histone interfaces, destabilizing the nucleosome structure. They can occur at the acidic patch, which is an exposed area on the nucleosome that's important for facilitating interactions between nucleosomes and effector proteins or nucleosomes and other nucleosomes. And they can also occur at other exposed residues, but generally this also leads to disrupted binding between nucleosomes and other proteins. They can also occur at histone DNA interfaces, again, destabilizing the nucleosome. And they can occur at those intrinsically disordered N-terminal tails on histones, leading to altered histone tail DNA binding or altered binding with effector proteins as well. Now, generally, these local effects are thought to generally scale to larger global effects, such as the perturbation and the regulation of DNA accessibility, chromatin compaction, DNA repair, DNA replication, gene expression as well, ultimately leading to oncogenesis. Now, while there have been a number of different histone mutations that have been demonstrated to drive cancer development, there are still hundreds more inside of cancer that have been observed, but we really don't know if these mutations are actually actively driving cancer development, and we don't know how they actually be driving cancer development. So really, they're just completely undercharacterized, and we don't really know the consequence of these mutations. So from this project, we have 3 key aims. The first is to investigate potential histone mutation oncogenic mechanisms. The second is to identify factors that predispose patients to developing histone mutations. And the third is to predict which histone mutations are actually actively driving cancer development rather than being passive byproducts of mutational processes. So in order to achieve this, what we've done is we've gathered whole exome sequencing data and clinical data for about 12,000 different patients. And these patients are spread across 68 different cancer types. One of these cancer types is actually a pan-cancer model as well that we've been using. And this data set also includes 2,800 different histone missense mutations. Now, one of the first things we want to do is actually determine how histone missense mutations might be contributing to oncogenesis mechanistically. So in order to achieve this, we've been using histone interaction networks, or also known as interactomes, and these were created by our collaborator, Dr. Young-Hui Peng, and they detail residue-level contacts between histone proteins and other biomolecules. They're based on structural data from the protein data bank, as well as cross-linking mass spec experiments. And from the protein data bank-based interactome, we define interactions as any instance where a histone-heavy atom, which is any atom except for hydrogen, was within 5 angstroms of another heavy atom from a binding partner. So it could be another histone protein, it could be DNA or another effector molecule. And from the cross-linking mass spec interactome, we define interactions as any instance where a histone lysine residue was able to form a cross-link with a lysine residue from another protein. And in the interactome, we've actually divided up the interactions into 3 different types. The first one being histone DNA interactions, the second being histone-histone interactions, and the third being histone other interactions, which are incredibly diverse. Basically, other partner refers to any protein that is not a histone. Now, one of the first things that we want to do is we want to determine where histone missense mutations localize with respect to nucleosomal interfaces, specifically histone-histone interfaces and histone DNA interfaces. And we want to determine if they're enriched or depleted at these interfaces. So we've done this for all mutations inside of our data set, inside of our pan-cancer model. And we've also done this for non-recurrent mutations as well as recurrent mutations, because we're interested to see if there's any difference between those two types, which could be potentially useful when predicting driver mutations as well. And so from this analysis, we found that histone missense mutations are enriched at H2A histone interfaces, as well as H2A DNA interfaces inside of the nucleosome, which is rather interesting. We haven't quite interpreted what this could potentially mean, but it is interesting to think that H2A histones might be, it could be debated that they're generally the most diverse structurally of all of the core histones. So there could be something there. We've also found that recurrent mutations are enriched at H2A DNA interfaces, as well as H2B histone and H3 histone interfaces as well. Now, we also want to determine where histone missense mutations associate with respect to those other interfaces that I previously described. Again, other being any protein that's not a histone protein. The only issue with the interactome is that not every single histone protein has the same level of evidence inside of it. However, histone proteins are generally structurally quite similar. So what we did is we performed a multiple sequence alignment. We mapped those histone other interfaces to positions on that multiple sequence alignment alongside histone mutations, and then try to determine if histone missense mutations are enriched or depleted at those aligned interfaces. And what we found was across all different histone types, generally speaking, histone missense mutations are enriched at histone other interfaces. And this is also the case for those recurrent mutations when we're looking at them specifically. And in contrast, we didn't find any significant difference for those non-recurrent mutations. So yeah, we're not really sure what the interpretation of this is yet, but we generally see that histone missense mutations like to overlap and diverse array of different interfaces for diverse array of partners. Now, previous literature has also shown that there's, generally speaking, a high frequency of histone missense mutations at acidic patch residues. However, it hasn't really been shown whether or not these mutations are actually enriched or depleted statistically at these interfaces or at these acidic patch residues. And just for a reminder, the acidic patch is actually an exposed area on the nucleosome that's responsible for facilitating a number of different interactions between nucleosomes and other proteins. And it's formed by a few different acidic residues on both H2A and H2B. And here it's actually interesting because we found that although there's a high frequency of histone missense mutations found at these acidic patch residues, we actually find a statistically significant depletion of missense mutations at these residues. Additionally, we also want to determine where histone missense mutations associate with respect to post-translational modification in sites as well, because previous results have shown that histone missense mutations also like to drive cancer development through perturbing post-translational modification signaling. And what we found here was that both recurrent and all mutations in general tend to be enriched at H2A post-translational modification sites. But interestingly, we found a depletion of recurrent mutations at H2A post-translational modification sites instead. Now, going back to the histone other category, the other category of histone binding partners, those partners are actually very highly diverse in function. And we want to determine if specific binding partners in histone other category may be targeted by histone mutations, consequently leading us to possibly discover certain pathways that histone missense mutations might be using to drive cancer development. So what we did is we have about a total of 366 different proteins inside this histone other category, and they could range in function from, say, histone chaperone proteins. It could be DNA transcription machinery or post-translation modification machinery. So what we did is we performed a series of binomial tests for each of these different partners, trying to determine if histone missense mutations associated interfaces that are specific for these partners. So what you can see here is kind of the results of those binomial tests inside of pan-cancer, with the full change on the x-axis and the inverse Q value or inverse P value on the y-axis. So it's just denoting statistical significance. Anything above the green line here in this plot is statistically significant. And we found a number of different partners or interfaces for specific partners that histone missense mutations generally like to aggregate to. And one interesting thing that I noticed here is that although we generally didn't find for histone missense mutations for a number of different histone types that histone missense mutations like to aggregate at either histone-histone interfaces or histone DNA interfaces, we did find that histone missense mutations like to aggregate at interfaces for specific histone chaperone proteins. So although histone missense mutations might not generally directly disrupt histone nucleosome structures, they might indirectly disrupt them through interfering with histone chaperones. Additionally, similarly, we also found that there's a number of histone post-translation modification readers, writers, and erasers that also are targeted by histone missense mutations as well. So although histone missense mutations might not directly be enriched or depleted at interfaces or at post-translation modification sites, they still may be interfering with post-translational modification signaling through disrupting the effector proteins that either deposit or move these different PTM marks. Now, histone interaction networks are incredibly useful for determining if histone missense mutations overlap with interaction interfaces. However, it can't exactly quantify how those mutations may affect those interfaces. So quantitatively, we can't determine if those mutations either disrupt or stabilize those interfaces. So we've used a couple of different machine learning methods to determine this, being SAMP3D and SAMP3D, and they're both machine learning based methods. So I won't really get into the details of what an XG boosted decision tree is. But what you do need to know is that these different methods basically predict the effect of a mutation on binding affinity. So basically saying how a mutation changes the binding affinity between a histone and another protein or histone DNA once it happens. And this is measured inside of kilocalories per mole, also represented as delta delta G. And the difference between SAMP3D and SAMP3D is that SAMP3D predicts specifically delta delta G values for protein-protein interactions, whereas SAMP3D predicts it for protein-DNA interactions. Now, unfortunately, not all the interactions that both of these methods actually require both a mutation and a structure. And unfortunately, a lot of the interactions that we have that we've annotated our histone missense mutations with might come from cross-linking mass spec data. And of course, we don't have any structures for these. So there are some instances where we can't actually predict how mutations affect an interaction, just because we don't have any structural data for that. But we still have managed to do about 200,000 different predictions, delta delta G predictions, for approximately 1,500 different mutations to see how these mutations might be affecting those interactions. Additionally, for some select interesting cases, we've also been using Mutavine 2 for additional validation in some situations. Now, one of the first things we want to do is we want to compare how both recurrent and non-recurrent mutations might differ in terms of their ability to disrupt nucleosomal histone-histone interactions. So here, what we can see is the distribution of delta delta G values for recurrent and non-recurrent mutations. Non-recurrent mutations are inside of red, and recurrent mutations inside of green. And we can see we've done this per histone type. In any instance where you see a delta delta G value above 0, this is generally referring to predictions where mutations actually destabilizing a histone-histone interaction. And anything below 0 are instances where a histone mutation is stable, or sorry, it should be the other way around. If it's positive, it should be destabilizing. If it's negative, it should be stabilizing. So just ignore the labels there, but if you see the red arrow, destabilizing, blue arrow, stabilizing. And we actually didn't find any statistically significant difference between recurrent or non-recurrent mutations in terms of these two different distributions, generally suggesting that both recurrent and non-recurrent mutations could be potentially equally as destabilizing. So there's no association there for histone-histone interactions. And similarly, we didn't find any association between recurrence for histone DNA interactions as well. And this is just from the results of a few different man-weighting U tests, but we also performed a few different correlations to try and determine, again, if the number of times a mutation was observed inside of our data set correlates with its delta delta G value. We didn't find any correlation there either as well. And although we didn't find anything interesting there, we have actually been able to integrate the delta G predictions that we got from those two machine learning methods alongside our histone interaction networks. And so basically what you're seeing here is a disruption model for our pan-cancer cancer type. So we have the different histone proteins colored here by histone type, green being histone H4 proteins, yellow being histone H2A proteins, blue being H3 proteins, and red being H2B. And then we also have in gray all those proteins that are belonging to that other category of histone binding partners. And each of these different lines that are drawn between a histone and a partner are actually representative of the mean delta G, or the mean disruption of mutations occurring inside of that histone protein with interfaces for that partner. And I've also colored these so that red lines denote that mutations are actually destabilizing interactions between histones and those partners. And then blue shows stabilizing interactions. And I'm sure, as you can see here, there isn't a lot of blue. So generally speaking, histone missense mutations tend to actually disrupt interactions between histones and these partners as well. And we've also are currently working on determining or identifying specific partners that histone missense mutations really like to disrupt. So for example, you can see fat complex subunit SSRP1 just over here is generally impacted quite heavily by histone missense mutations. Now, as part of our project, we also wanted to identify which patients might be specifically prone to harboring histone missense mutations. And the first place we decided to look was actually determine if cancer type is associated with the presence or absence of histone missense mutations. And so we actually did manage to find a number of different cancer types where histone missense mutations occur more frequently when compared to patients inside of other cancer types. And so here, what you can see is the results of a few different Fisher's exact tests. And on the x-axis, we have a number of our different cancer types shown, as well as an odds ratio showing whether these cancer types are enriched or depleted for patients with histone missense mutations. And so all of those cancer types with a blue dot show instances where they're enriched for patients with a histone missense mutation, and all those cancer types with the red dot show instances where those cancer types are depleted for those patients. And so, yeah, we have managed to find a few different cancer types that are enriched for patients with a histone with a higher frequency of histone missense mutations. But it is possible that one thing that we noticed is that it is possible that maybe the reason that these different cancer types are enriched for patients with histone missense mutations is because of overall higher tumor mutational burdens. So it might not necessarily be that these mutations are actually playing an active role in driving cancer development inside of these different cancers, but it could just be the byproduct of increased tumor mutational burdens inside of these cancer types. So another thing that we did was we also wanted to determine which cancer types generally exhibit higher tumor mutational burdens in comparison to other cancer types. And here I've just shown all the instances where we find either statistically significant enrichment or depletion in terms of tumor mutational burden. Similar to the last plot here, we can see actually with those blue dots, different cancer types that exhibit higher tumor mutational burdens in comparison, turn this off, in comparison to other cancer types. And then in red here, we also have those cancer types which have lower tumor mutational burdens. And interestingly, we found that all those different cancer types which exhibit higher frequencies of patients with histone missense mutations also exhibit higher tumor mutational burdens, which really begs the question, is it that tumor mutational burden or higher overall tumor mutational burdens are causing histone missense mutations to occur at a higher rate inside these cancer types? Or are these histone missense mutations potentially leading to higher tumor mutational burdens? That's a question that I'm going to address a little bit later. But yeah. Now, because we did see that, the frequency of histone missense mutations could vary by cancer type, we also want to determine if basically histone missense mutations generally behave differently inside these different cancer types as well. So going back to those binomial tests that we saw, that I showed before, where we were trying to determine if histone missense mutations associate with interfaces for specific partners, we've also repeated these tests within individual cancer types. in order to determine inside of a specific cancer type if histone missense mutations use specific pathways there to drive cancer development. And what we've done is we've also found some interesting common trends between different cancer types where there are certain proteins that are generally targeted by histone missense mutations in multiple different cancer types. And here we have an example of 3 of those proteins that are targeted across several different cancer types, actually. DNA replication licensing factor, MCM2, histone chaperone, ASF1A, and tonsoku-like protein. Now, what I found interesting about these 3 different proteins is that actually a number of the cancer types that we found to have a higher tumor mutational burden and a higher frequency of patients with histone missense mutation, we also found that the mutations inside of these cancer types generally tend to aggregate interfaces for these proteins. And the reason that I find this so interesting is because these proteins are actually part of a specific pathway for double-stranded break repair. So MCM2 is actually a component of a helicase enzyme, which just unwinds DNA. And then once the replication fork encounters a double-stranded break, histone chaperone ASF1A actually deposits new histones, H3 and H4, to that area of that double-stranded break. And then in turn, tonsoku-like protein, or tonsil, is able to recognize unmethylated K20 residues on that H4 protein and recruit RAD51 for homologous recombination to occur. So it is possible that a lot of these histone missense mutations are targeting this pathway, consequently leading to less homologous repair and potentially even leading to more non-homologous end joining, leading to overall higher mutation rates. So yeah, that is one potential reason why we see that those different cancer types with higher frequencies of histone missense mutations might also have higher tumor mutational burdens, although not 100% certain yet that that is the case. Now, on to driver prediction. So typically, clinically, frequency is generally used quite often to actually predict whether a histone or any mutation is actually driving cancer development or not. However, there's one critical issue with this, and that kind of goes back to what we were discussing about with the tumor mutational burden, is that we might see a high frequency of a given mutation just simply because there are a lot of different factors driving that mutation to occur more often inside of that cancer type. So what mutagene does is it actually normalizes the observed frequency by an expected mutation rate for every single mutation inside of a given cancer type. And the reason that we chose this method is because it performs competitively with other methods, as well as providing us easily interpretable results. And the results that it actually provides are B-scores, which is basically like saying, what is the probability of observing a mutation at the observed rate given some sort of background prior expectation for this mutation to be, or background prior probability for this mutation to be mutated at whatever given rate. And from this B-score, we're actually able to divide using 2 different thresholds to divide mutations into driver mutations, potential driver mutations, or passenger mutations. And just to give an example of kind of like how this looks, this is actually our rankings for histone missense mutations inside of, I believe, multiple myeloma. And I've colored mutations by their mutagene label. So red being drivers, yellow potential drivers, and green passenger mutations. And on the x-axis here, we have the total number of observed mutations, or the number of times that mutation was observed inside multiple myeloma. And on the y-axis, we have inverse or negative log of the mutability, or the expected mutation rate for that mutation. And we see that, generally speaking, as you increase the number of observed mutations, it's more likely that that mutation will be predicted as a driver, while also, so if that mutation has a higher number of observations but a lower mutability, then it's more likely to be a driver. If it has a higher mutability but a lower number of predictions, then it's more likely to be predicted as a passenger. So yeah, all we're doing is normalizing the observed rate by some sort of expected prior. And so we've performed mutagene driver prediction for each histone mutation inside of all our cancer types, except one thing that we did beforehand was we actually filtered out patients that were considered outliers in terms of tumor mutational burden, just because this can skew some background models when we're performing or doing our driver prediction. And And consequently, this has led to a loss of us not being able to predict a couple mutations as drivers or passengers or potential drivers. So from the 2,800 mutations that we initially started with, we were only able to predict whether about 1,600 of these mutations are actively driving cancer development or not. And then after our driver prediction, we've managed to find a total of about 219 different driver mutations spread across 58 out of our 68 different cancer types. Now, one interesting insight about these driver mutations is that generally speaking, they seem to be very specific to individual cancer types, with only a very few of them actually being predicted as drivers in multiple different cancer types. And so here I'd just like to highlight some instances where a mutation has been predicted as a driver inside of multiple different cancer types. So here on the x-axis, what you can see are 6 different mutations. And on the y-axis, the cancer types that are predicted as a driver in, or in some cases, a passenger, potential driver, or weren't observed in, inside of gray. And we've also shown the number of times that that mutation was observed inside of that cancer type, inside of each cell, as well as the B-score for mutagene. And interestingly, we actually see that we found about 3 different mutations that have actually been previously described inside of the literature and predicted those to be drivers inside of multiple different cancer types. And I'm not really sure if anybody was able to visit Dr. Nadjabato's seminar recently, but these are actually the 3 different mutations that she was describing, which have been previously found to occur inside of pediatric glioblastomas. However, inside of this instance, we've actually managed to find that these mutations may be acting as drivers inside of adult glioblastomas, as well as additional other different cancer types as well. So in addition to some of these previously described mutations, we also have found a few different novel mutations as well that have been predicted as drivers in multiple cancer types. And that includes the H2BC21F70L mutation. This mutation has been previously found to occur at a high frequency inside of pan cancer. However, it has not actually been directly described as a driver mutation yet. And within our data set, this is actually still the most frequently observed histone missense mutation that we have. And through using our information from the histone interaction networks, as well as delta G prediction methods, we found that this mutation is actually able to disrupt histone-histone interactions inside of a hydrophobic pocket deep within inside of the nucleosome. So a potential mechanism for this mutation is that it could actually be disrupting nucleosome structures. Now, another mutation that we found is the H3.1E133Q mutation, and which is rather peculiar. We're still looking a little bit into this mutation, but it's interesting because it occurs on the C terminus of the H3.1 protein, where generally speaking, there haven't been any other driver mutations that have been found inside of this region before. And then finally, we also have the H4R67P mutation, which was found as a driver inside of 2 different cancer types. And this mutation is actually particularly interesting because it occurs inside of an H4 histone. And H4 histones are actually probably the most well-conserved protein overall. They really, evolutionarily, they only differ by a few different amino acids. And there really haven't been any mutations that have been previously predicted as drivers inside of H4 proteins as well. Now, one other thing that we've been able to do is we've been able to integrate our results from mutagene, our driver prediction method, alongside some of the information from our histone interaction networks. And this is going back to those binomial tests. What we've also done is we've determined whether specifically histone missense mutations of a particular label within a specific cancer type are enriched or depleted at interfaces for specific proteins. So here, what I basically shown is just those binomial tests again, just performed inside of squamous cell carcinoma. But in this instance, we're only considering mutations of a specific class. So that could be mutations that have been predicted as drivers in squamous cell carcinoma, those that have been predicted as potential driver mutations inside of squamous cell carcinoma, or those that have been predicted as passengers. So we're still trying to look at a few of the results here to try and see if there's any differences between the partners that driver mutations, potential driver mutations, and passenger mutations like to tend to target. But one interesting thing that we have found is that in comparison to potential driver mutations and passenger mutations, the fold change of driver mutations at interfaces for specific proteins is actually quite large. So for example, here we have metal response element binding transcription factor 2, and that we found that in comparison to the number of expected mutations at interfaces for this protein, we have about a 700-fold change. So this could potentially be one protein or part of a pathway that is really targeted by histone missense mutations to drive cancer development inside of this specific cancer type. And another thing that we did actually prior to mutagene ranking, determining whether histone missense mutations are drivers, potential drivers, or passengers, is we want to see if there are any mutations which are hyperdisruptive outliers of histone-histone interfaces and histone DNA interfaces within inside of the nucleosome. So here we can actually observe a few different mutations which have been predicted as hyperdisruptive outliers of histone-histone interactions within the context of the nucleosome. So those are all shown on the x-axis here. And we also have the distributions of their delta-delta G values for those histone-histone interactions on the y-axis. And just for reference, I've also provided the delta-delta G distribution of all mutations together on the far left here on this plot. And interestingly, when we actually look at this plot alongside of our mutagene results, about 7 out of 10 of those different mutations that we predicted that we saw to be hyperdisruptive outliers of histone-histone interactions within the context of the nucleosome have been predicted as either a driver or potential driver within at least one cancer type. Now, comparatively, the other 3 mutations that we did not predict as a driver or potential driver inside of a given cancer type were actually removed prior to driver prediction just because they occurred inside of patients that had too high of a tumor mutational burden. So yeah, this leads us to think that these not only are these mutations act potential drivers or drivers of these specific cancer types, but they also might act through disrupting nucleosome structures as well. And another interesting thing is that mutagene doesn't consider any structural information at all. It's basically just all based around observed mutation rates, and there's expected prior mutabilities. However, here we've also found that these mutations structurally or their effects on nucleosome structures are quite distinct from other mutations as well. And one really interesting thing here is that we actually found that H4R67P mutation that I previously described as acting as a driver in multiple different cancer types. And this is also found to be hyperdisruptive of histone-histone interactions within the nucleosome structure. So additionally, we are investigating whether this mutation could potentially be driving cancer development through disrupting nucleosome structures, or if it actually acts through a few different other mechanisms, because we have found this mutation to be able to disrupt interactions with BRCA-associated ring domain 1. And here we can see in the top right, the H4R67 residue interacting with that protein inside the context of the nucleosome. It also disrupts interactions with MCM2, as well as a histone chaperone protein, death domain associated protein, or DAX. Now, so through this project, we've actually been able to identify a number of different histone missense mutations, predict them as drivers, see where they associate with respect to interaction interfaces, and determine whether or not these mutations are actually disrupting these interfaces or not. However, there's only so much that we can do as far as characterization in that regard, just using the methods that we have available. So we have been collaborating with a number of people, and we also have a number of different members inside of our lab that are also working with these different histone missense mutations to further characterize them as well. And so here I'd just like to show some of the work that Dr. Shuzheng Li, a research associate inside of our laboratory, has been doing. And what he's done is he's actually been performing a few different physics-based simulations of these mutations within the context of the nucleosome to determine how they actually differ, how they affect sorry, let me see if we can play this. So how they affect interactions in these simulations. So here, where's the cursor? We can see the G107 residue, or the absence of it because it's a glycine, probably the least exciting amino acid, and how the H2B protein interacts within the context of the nucleosome with H4 neighboring H4 protein on the right here. And then, 1 second. And then upon mutation, this is another mutation that we predict as a driver inside of at least one cancer type inside of our data set, the H4G1 or H2B G107R mutation. When this mutation is actually turned from a glycine into a arginine residue, we see that the H2B protein actually starts interacting a lot more with the H4 protein, potentially stabilizing the nucleosome, leading to unknown effects so far. But it is interesting to see that these mutations have been shown to actually have an effect, at least inside of an in silico model. Now, another thing that we've also been collaborating with Dr. Maria Stizabal from the biology department to kind of determine how these mutations might work inside of different model systems, specifically inside of a yeast system. And here I'm just showcasing some of the work of master's student Austin Macklem from the Stizabal lab. And what you're seeing here is a panel of different yeast that Austin has actually transformed with homologs of mutations found to be drivers as drivers inside of our histone mutation data set. And he's actually managed to determine that while a lot of these mutations are viable inside of yeast cells, depending on the conditions, that these mutations can actually lead these yeast to exhibit growth defects. So it's interesting to see that inside of these model systems that these mutations are actually affecting cellular phenotypes. And so we're looking forward to actually furthering the investigation with Dr. Stizabal to see how else these mutations might affect phenotypes. And in addition to looking at H2B mutations, we're also looking at H2A mutations and mutations inside of specific histone variants as well. So for a quick recap, basically what we've done with this project is we've gathered whole exome sequencing data as well as a clinical supplement. And alongside the use of histone interaction networks, structure-based delta-delta G prediction, and cancer-specific driver prediction, we've managed to determine how histone missense mutations associate with interaction interfaces, how they might disrupt these interaction interfaces, which mutations might be actually acting as drivers inside of a specific cancer type, and determining which specific cancer types may actually be more prone or less prone to harboring histone missense mutations. So 3 key points, if you didn't get anything from that presentation. I would say that number 1, the prevalence of histone, from this project, we've determined that the prevalence of histone missense mutations in cancer may be substantially higher than previously thought. And this actually prevalence varies quite a bit by cancer type. So inside of our data set, we found about 16% of patients to harbor a histone missense mutation, although previous reports suggest that only about 4% of patients harbor histone missense mutation. Second is that while there are thousands of histone missense mutations in cancer, only a few of these may actually actively drive cancer development, and this does again vary by cancer type. And third, histone missense mutations may drive cancer development through using very specific mechanisms, also on a per cancer type basis. So with that, I would like to thank my supervisor, Dr. Anna Panchenka, and our collaborator, Dr. Maria Stizabal, David Landsman, Yang Hui Peng as well, and members of the Panchenka Laboratory, including Dr. Xiuzhang Li and Daria Ostrovakova, as well as Austin Macklem, for providing those beautiful figures of the yeast that we had previously shown. I'd also like to thank the Center for Advanced Computing for making this work able to be providing the resources for this work, as well as all the different patients and organizers from the programs that we use to gather this data from. And yeah, with that, I'll take any questions.
Thanks. So if people have questions on the Zoom, you can either use the raise hand function or just start yelling out. We'll hear you. I'll see it if you use the raise hand. So questions from the audience. Yeah.
I'm just curious, are any of the histone mutations hereditary or are they all...
Yeah, so these are all somatic mutations. We would have liked to do germline mutations, except we just didn't have that data available. So yeah, all these purely somatic.
But are there any germline or heritable mutations?
Not to my knowledge, like previously reported in literature, I haven't heard of any germline mutations. All of them that have been shown to be drivers have been somatic so far. But I mean, I'm certain that it could be possible. Just I don't have that information. on hand.
Well, it kind of leads into one of my questions, which is, what the magnitude effect of these mutations that you're looking at. So I'm assuming that it happens in the context of lots of other mutations in these tumors.
Mhm.
So, in comparison to, I don't know, a KRAS G12 to V or something, one of these very powerful oncogenes. What do you think the magnitude effect is going to be? That's a tough question. I know.
Yeah,
But linked to that is, do you see any other cooperating mutations that are more frequently
Yeah.
observed? Yeah.
yeah, so that's actually one of the things that we're interested in looking into is like the co-occurrence between histone missense mutations that have been produced as drivers and other mutations inside of other, genes that could also be potentially driving cancer development. And we're also trying to look into whether these histone missense mutations could be acting synergistically with other mutations.
Right.
And that's like, it's totally possible. I mean, on average, I think cancer types generally have, for each cancer, there's about 2 to 4 different driver mutations. So they're not actually acting, in isolation or alone. They could be acting synergistically together.
So that's an interesting question, to say that there's 2 to 4 driver mutations in a cancer. Because my understanding is when people do the whole genome sequencing, it's not uncommon to see tens of thousands of somatic mutations.
Mhm.
So what you guys are doing are trying to sort through all of that to try and identify driver mutations. So is that where you come up with an estimate of 2 to 4 driver mutations in a tumor?
No, that's actually what I've seen previously inside of literature. Yeah.
So is that how those numbers are arrived at? Is that kind of analysis? And I guess it gets back to magnitude effect, because people can do things like introduce, I don't know, a very specific, powerful oncogene into a cell line, and then stick it in an animal and see tumors arise, and or more often, I think that work was to actually make transgenic animals so that the animal went through development, and presumably what's happening along the line is as development proceeds and the animals reach adulthood, it's later on that they develop tumors. And then when you go and look at those tumors, there are lots of other mutations besides the mutant oncogene that you put in there. So do histone mutations occur in those contexts, do you think? So, and it gets to your point about synergy. So is there some indication of how specific histone mutations might synergize with specific oncogenes?
Yeah.
I guess it kind of came full circle.
Yeah,
Question again.
Like I haven't really heard of any, synergistic interactions between histone mutations and other mutations previously. Although, I generally wouldn't be surprised if that's something that we should probably look into that we would see in the future. Yeah, like there have been a few different histone missense mutations which have been shown to actually potentially increase tumor mutational burden, which is kind of also what we see here as well. Yeah, I'm so sorry.
Well,
Does that answer your question?
Yeah.
Or
I think I get the sense of where this is. But I guess the magnitude effect of any given histone mutation is probably, at least I haven't seen it in literature where people have
whether it's like certain to
targeted,
drive cancer development, if
germline
induced.
Make a zygote mutation.
Yeah, she's done that with, yeah.
And she's also done it in cell lines and they get transformed.
Mhm.
But it's
But
some of
so defects in development, but progression to malignancy,
I think they get so messed up that they had to.
don't make it through development. Then. So that would suggest a very high magnitude of impact.
Yeah, it does depend on the mutation as well. Like I think that was specifically.
Like one of the mutations that you do develop.
I think that was G34 mutations inside of H3 proteins. Yeah, and they have shown that before, but I think that also like the magnitude of oncogenicity could also vary by cell type as well. So for example, like a lot of those mutations that Dr. Jabata was talking about inside of her seminar, were only contextually drivers inside of um, very specific cases of glioma, pediatric high-grade gliomas, I think in the midline of the thalamus, whereas you don't really observe them inside of any other cancer types, just because in those cancer types, it actually wouldn't actively be driving cancer development, just because these mutations aren't just, it's not all completely genetic based how they actually might be driving cancer development. There's a large epigenetic component to it as well. So yeah, like how it influences post-translational modifications, histone proteins can lead to different cell fates. It could, take on different cellular phenotypes, potentially regress into more of like a stem cell state, and then progress to malignancy. So it also like really depends on, the cell type as well that we see these histone mutations inside of.
Thanks. Chris Nickel has a question. Go ahead, Chris.
Thanks, Peter. Thanks, Daniel. Great talk. Along the lines of what Peter was asking, have you looked, and maybe if I missed it, I apologize. Have you looked to see where, or in the subtypes of various cancers that have a high genetic instability component to them, if some of these histone mutations are some of those additional early events that help to drive those types of cancers?
Yeah.
Just in terms of, you talk about some of them are drivers, but I'm thinking as, I think similar to Peter, is it a chicken and egg thing? Do these come first and then others happen later, or do some of the initial ones come first and then if you magically hit some of these histone mutations, you get the types of cancers that have the higher genetic instability, et cetera, et cetera?
Yeah, that's actually a little bit difficult to determine given the data we have, because a lot of these patients only have like one tumor mutational or tumor sample. So like we can't really, compare how these mutations differ over time, to see if, that mutation, that histone missense mutation occurred early on inside the tumor's development. And then if other mutations happen afterwards, or if there were, specific mutations that led to that histone missense mutation occurring in the patient later on. It'd be nice if we had, a number of different tumor samples per patient over a period of time, but I just don't think we have that information. At most, we might have 2 or 3 different mutation or tumor samples for a given patient. So, but yeah, it's an excellent point. That'll be really useful in determining whether, the whole chicken and egg theory, is it the histone missense mutations that are actually causing higher tumor mutational burdens to occur, or is it a higher tumor mutational burden that is leading to histone missense mutations appearing?
At your point on binding, I think will explain sort of what is a chicken and egg.
Yeah.
Did you say binding?
Yeah. Yeah. So, I mean, we did show that, histone missense mutations generally like to target specific proteins that are involved inside of repairing DNA or DNA repair pathways. Like I still can't say with 100% certainty based off of that analysis that, it's definitely the histone missense mutations that are driving cancer development, but it does give us at least a solid lead to kind of go off of and investigate in the future.
You can say we're doing it in yeast.
Yeah, you can do it in yeast.
A little bit of data that says big cost for damage.
Interesting. Okay.
Yeah.
That's cool to hear.
Go ahead, Gabby.
Great presentation. I actually have 2 questions with that.
Yep.
Firstly, the difference for canonical versus variant histones.
Yeah. Yeah.
Repeat that question for the audience.
Yeah. So the question was, first to reiterate the difference between variant and canonical histones, and then kind of touch on whether, is it driver mutations or just mutations in general? Whether driver mutations occur more frequently inside of variant histones versus canonical histones. So for the first question, canonical versus variant histones, they differ in quite a few different ways. The first one's expression timing. Canonical histones are only really expressed during G1 and S phase, whereas variant histones can actually be expressed at any points out of the cell cycle. The second one, although not a defining characteristic, is that variant histones are generally more structurally diverse. Canonical histones may differ from each other within that same group by maybe 1 or 2 amino acids, whereas variant histones, you can see some of them differing from each other by entire domains. More often than not, it's only a few amino acids, but that can be the case. There are some other different features inside of the mRNAs between canonical histones and variant histones as well. I'm not gonna, if you want me to, I can dive into that, but it gets a little bit deep. They do use different pathways.
Wild type and mutant type is like.
Sorry?
Can we call the canonical mutant variants as like wild type histone and the variants are mutant type?
No, so you're thinking like variant in the sense of like a mutation. Histone nomenclature kind of admittedly sucks. I'm not gonna lie. Because for a typical protein, you might say like anything is like a variant, and it might be like one flavor or another of a specific protein that belongs to a specific family. Histones are kind of weird because we have these 2 different camps. One of them is called variant histones, the other one's called canonical histones, and they differ basically by the different characteristics that I'm describing now. So again, being the replication dependency of canonical histones, structural feature variation inside of variant histones, as well as variant histones also use some different mRNA processing pathways. And genomically, they're also quite dispersed, whereas canonical histones are actually clustered in pretty much the same genomic region. Generally, that's thought to be because the cell needs to produce a lot of canonical histone proteins during S phase, so it just kind of likes to be in the same area. Yeah, so there's a few different differences. I think, have I answered your question?
Did you get all your answers there, Gabby?
Not quite yet. Yeah, I had looked into that previously, but we've had some changing around our data set and our analyses. So I can't say like 100% for certain whether or not, variant histones might harbor more or less mutations than canonical ones. We have looked at the individual proteins themselves, regardless of whether they're canonical or variants, and we have found that certain histone proteins actually really, I wouldn't say like histone mutations, but they generally accumulate a lot more in comparison to others. So that's another thing that we're still exploring. Yeah.
Sorry, one follow-up question, and different topic, but
Mhm.
it was really interesting how you went into its role in the double-strand break.
Yep.
And something that we work on in our lab is focusing on BRCA1 and its DNA double-strand break, very accessible missense mutations in that gene, how it affects its ability to do that function.
Mhm.
So something that we're employing are inhibitor sensitivity assays where we induce single-strand breaks in DNA, introduce like and then see how it would react such that if BRCA1 is sufficient enough that it can conduct double-strand break repair, or if the cell will die because there's insufficient BRCA1.
Mhm.
Do you think this could be a potential future direction for assessing whether a histone does in fact disrupt proper DNA double-strand break repair and possibly certain therapeutic implications?
Yeah, I mean, it's definitely 100%. Yeah, I think that'd be something interesting to be looking into. I'm a computational biologist, so Austin, if you'd like to do that for me, I'd be very appreciative. Yeah.
Go ahead, you're first, then Matthew.
Have you looked at where the histone mutations that are in histone other protein interactions, if the region where they're interacting with on the other protein, if that can be mutated and also be a driver?
Yes, actually, we have. I don't have the results on hand now. We have found a number of driver mutations inside of those proteins that those histone mutations target, but we haven't found them exactly at the interfaces that those histone mutations occur in. So, I mean, could be driving cancer development out of those proteins through other mechanisms, not necessarily disrupting interfaces between histones and those proteins. But it is interesting to see because, regardless, that protein still plays the same role, has the same function. So if we see a driver mutation inside of like that specific protein, maybe, and that through a certain pathway it's driving cancer development, maybe that also gives it a little bit of evidence that our histone mutation could be kind of hijacking the same pathway to drive cancer development. And actually that would be something that would be interesting to look into as far as mutation co-occurrence goes as well, to see, you know, if there's patients that generally tend to have mutations inside of a histone and a given protein, or if it's mutually exclusive between the two. Yeah.
Great talk. You had a nice figure or diagram that showed that multiple genes can come from the same.
Yeah.
Right?
Yep.
Doesn't matter which gene is hit, is it a dosage kind of thing?
Yeah.
Yeah.
Yeah, that's like probably one of the most difficult things about working with histone mutations, is that there's such a high redundancy, functional redundancy, and especially in terms of both structures and genes as well. It's really hard to speak to that without like doing further characterization experimentally. There have been mutations in the past that occur inside of on one specific gene that encodes a given protein, but where there's other genes that can also encode for it as well. And they have been shown to actually drive cancer development inside of a dominant fashion. And you know, this took years of work to actually determine and figure out that through like a very specific mechanism. So it's 100% possible. Just right now, off of what we have, I can't exactly say whether that would be the case for all of our mutations. But for some reason, certain histone genes generally, even if they encode for the exact same protein, might tend to accumulate more mutations than others, which is a little bit peculiar. It could be because of codon bias. There might be certain codons that could be mutated more often than others inside of a given gene versus another gene that might encode for the same protein. And that just might be the reason why we see more mutations inside of it. Yeah. It's difficult to say, like, for any individual case, whether, you know, that mutation is going to act in a dominant fashion or if it's going to be recessive, so to speak. So
that sort of stimulates another question that I had. It has to do with the stoichiometry of canonical versus variant histone. So I'm guessing that, well, tell me, what is the stoichiometry in the histone population in the cell? Do you have, are the histones more likely to be made up of more of the canonicals?
it varies. It really varies by cell
And
type.
how does it also vary by cell cycle or differentiation state, other things?
Yeah, I imagine it would. I'm not 100% certain. But, you know, again, canonical histones are very, very rapidly produced during, you know, G1 and S phase.
So, and is that just because the cell needs twice as much histones, presumably, and those are the genes that are more likely to get the canonical ones to get upregulated more rapidly?
Yeah, so you know, during S phase, you're producing a ton of DNA, and you don't want to kind of just be everywhere inside of the cell because it can, you know, lead to a lot of different issues, DNA damage, infidelity inside replication. So you need a lot of histones to be able to actually come in and protect that DNA. And which is actually why it's also expressed a little bit inside G1 phase. It's kind of like a loading phase for histone proteins to kind of start being produced before DNA comes out so that any new DNA can be protected. Sorry, I got a little bit off track there, but yeah, canonical histones, basically the difference between that and variant histones is canonical histones cluster inside specific regions of the genome so that any expression machinery can rapidly produce histone proteins and very, very quickly, you know, kind of, I guess, sequester, I want to say, almost the DNA from any potential agents that could damage it. And then variant histones more dispersed throughout the genome.
Are you talking about the histone proteins or the histone genes? Because I think that's an important distinction.
Yeah, well, I'm thinking about the stoichiometry of the histones in the nucleus, in the structural components where they're sitting.
Protein or the genes?
The protein. So I'm
protein.
talking about
yeah.
the
Because I think you're
clone,
talking about the genes themselves.
Yeah, the genes would lead to the protein, but the genes are clustered so that a lot of these proteins can actually be produced in a very short period of time. So like after S phase, shortly during S phase and shortly after S phase, you might expect more canonical histones to be inside of
Has
them.
anybody been able to look at whether the stoichiometry, the sort of the ratio of canonical to variant histones, has any effect on whether that DNA is transcriptionally active or not? Does it correlate with cell? It presumably correlates with cell cycle because of that upsurge of canonical histone production. And I'm guessing that graph you showed us is protein. It's not.
Yeah, it's histone proteins. To answer your question, more or less, yes, there are like certain histone variants that could actually be indicative of transcriptionally active areas. I'm not entirely sure which ones off the top of my head would indicate that. But again, like, you know, they're structurally, they vary quite a bit. So it could actually, some of them might not bind DNA as well. So making it more accessible to effector proteins to come in and transcribe. And again, like other things that could indicate whether something's transcriptionally active or not could also be histone push translational modifications. Yeah.
Thanks. Other questions? Okay, we've had a nice discussion. Join me in thanking Daniel for a very nice talk. That's